View typical of Anatropous ovule. When the funicle, Chalaza, and micropyle, they are not present at a straight line. Just look at what I'm saying. I'm saying when the funicle, Chalaza, and micropyle, they are not present into the straight line. That is called the Anatropous ovule. That is called the Anatropous ovule. Now you can see this is the Chalaza. If you draw the straight line, we are getting micropyle, but funicle is not here. Funicle is present at 180 degree. Chalaza and micropylar. Chalaza and micropylar pole both are in straight line, but funicle is the at 180 degree. Clear? So such type of structure is called the Anatropous ovule. Anatropous ovule, where the micropyle and Chalaza, both are in straight line. Just look it, I drawn here. Look it again. This is the micropylar pole, micropylar end, and this is the chalazal pole, chalazal end. If you draw the straight line, you will get into the straight line. But funicle is here, funicle not here. So in this way, you can say funicle present at 180 degree. If you draw, so funicle present at 180 degree angle, means 180 degree. Clear? In this way, you can say this is a Anatropous ovules where the micropyle and chalaza present on a single plane and the funicle present at 180 degree angle. Okay. So means in Orthotropous, I told these three structure means micropyle and chalaza, funicle. These three structure present in straight line. That is called Orthotropous ovule. Orthotropous. Okay. And then inside they have this is the egg cell and two synergid cells. Egg cell, and here what is happening? Synergid cells. This is the synergid cells, and antipodal cell is there. This is the antipodal cell, and in center they have the nucleus, means double cell. Okay, now it is correct. This structure is known as the micropyle. So this is the micropylar end, and this structure is known as the chalaza. This structure is known as the chalaza, and you can see the outer. This is the integument. This is the structure which is known as the integument, and this integument, it is known as the outer integument. This structure is known as the outer integument, and this structure is known as the inner integument. This structure is known as the inner integument. Clear? Outer integument, inner integument. Okay, and this is called the chalaza, and this is the micropylar end. Okay, and this is the funicle. Funicle, I told funicle is the stalk-like structure. Funicle is the stalk-like structure by which it is the location. Funicle is the stalk-like structure. Just look at this is the stalk. Okay, funicle is the stalk-like structure. Wait once. Funicle is the stalk-like structure by which ovules gets attached with the placenta. Funicle is the site or location, and it is the stalk-like structure by which ovule gets attached with the placenta. Is it clear? Is it clear to all of you? Is it clear to all of you?
Yes, sir.
Yes, sir.
Okay, very good. So this is the structure. Now you can see same structure which I have written here. They have given. That is the chalazal pole. This is the chalazal pole. Here you can say chalazal pole. Then another is the embryo sac. This structure is known as the embryo sac. This structure is known as the embryo sac. So here embryo sac is there, and this wall, the wall of this, they have the special type of structure which is known as the nucellus. So this structure is known as the nucellus. Okay, nucellus. This structure is known as nucellus. So now here you can say nucellus. Outer integument, that means inner integuments, inner side. Outer integuments, outer side. Micropylar poles, here I drawn micropylar pole. and funicle here I drawn, and Hillum is the location. This structure is called the Hillum, where the ovules gets attached with the funicle. So this structure is known as the Hillum you will also you have also studied into the uh, kidney, whereas also Hillum found. Okay, so this is the structure which I have drawn, and here already drawn in your NCERT book, through which you can compare, which I have drawn. That is the same thing. They have inverted represents. This is also called the inverted ovule, because inverted represents, and here they represents like that. And here they represents like that. So both are the same thing. Are you getting on it? If we'll tilt this
Sir.
structure, if suppose that I'm going to tilt this structure in this way, so then if you tilt in this structure, so you will get same structure which I have drawn here. Clear? So this is the structure of and wheat. They have the single ovules, and papaya, watermelon, and orchids, they have the more than one ovules. Clear? Now another structure, that is the ovules, you can see. Let us familiarize ourselves with the structure of typical angiosperm ovules. The ovule is a small structure attached to the placenta by means of a stalk. I told funicle. Funicle means a location where the stalk-like structure. Funicle means a stalk-like structure by which ovules get attached with the placenta. What is the funicle? I'm writing again. Just look it. Funicle. Funicle is the a stalk-like structure or location, a stalk-like structure by which ovules gets attached with the placenta. That is called funicle. Okay, so here they again, the structure attached to the placenta by means of stalk, that is called stalk-like structure called funicle. The body of the ovule fuses with the funicle in the region called hilum. Okay, what is the hilum? Hilum I told also, it is the junction. Where ovules attached with just clear your concept. Okay. Mhm. It is the junction where ovules attached with the funicle that is called the hilum. I'm giving one homework. Just tell me what will be the correct. I'm showing to you this figure. Can you tell the exact position of hilum? Oh, they have already given hilum position already given. Don't find it. Encircle the nucellus except at the tip where the small opening called micropyle. If you'll see that these ovules, they are surrounded by the integument, but some places where the integument is not present there. But some places there is no integument, and that place, you can say that place is known as the micropyle. Micropyle is the small opening where no integuments, no covering is found. So just read this sentence and clear your concept. It have given each ovule one or two protective envelope called integuments. Integuments encircle means surrounds the nucellus except at the tip where small openings, that is opening is called the micropyle is organized. Means that integuments, they cover throughout the nucellus, throughout the ovules, but except somewhere where is the micropyle, that is a small openings found, which is known as the micropyle. There is no integuments is present. Okay, where is no integuments only present. Clear? Okay, let's look it. Opposite of the micropylar ends, that is called chalazal end, represents the basal part of the ovule. Okay, that is called the basal part of the ovule. Are you getting on it? Chalazal end is the Chalazal end is the basal part of ovule. Basal part of ovule. Chalazal end is the basal part of ovule. Clear? Is it clear? Micropyle, chalaza, hilum, funicle, integuments, everything is clear. Everything is okay. Shreya, okay, wa?
Yes, sir.
Very good.
Yes, sir.
Now, again, enclosed within the integuments is a mass of cells called nucellus. Okay, they have, I told already, nucellus, and cells of the nucellus have abundant reserve food material. Means if the cells have abundant reserve food material, means what is the function of nucellus? It will provide the nutrition because they have the abundant reserve food material. The basic function of the nucellus, it provide the nourishment. Okay, located in the nucellus is the embryo sac or female gametophyte. It is found into the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from the megaspore. I told after the means during the megasporogenesis, 4 linear megaspore tetrad form. 4 linear megaspore tetrad form. Out of 4, 3 become degenerate, 1 will become function. That is called the functional megaspore. That structure is known as the functional linear megaspore. Is it clear? And that megaspore is going to formation of the embryo sac or female gametophyte. Okay, it will form embryo sac or female gametophyte. Is it okay? Is it fine? Is it clear to all of you? Shreya?
Yes, sir.
Okay.
Sir,
Now you can see. Again what it have given? Megasporogenesis. I told already megasporogenesis story. That's is very simple story. You can see. That story is very simple. The process of formation of megaspore from the megaspore mother cell called megasporogenesis. Ovules generally differentiate a single megaspore mother cells, the micropylar region. I told this concept. I'm going to revise again that concept. You can see. Just wait. Okay, this figure representing, look it. This is the figure representing how the formation of embryo sac, not megasporogenesis. Okay, they are going to represent the embryo sac, how embryo sac or female gametophyte is going to form. Megasporogenesis, I'm going to revise again. I told primary archisporial cell, primary archisporial cell, archisporial cell, archisporial cell. It will divide into two. They form the primary parietal cells and sporogenous cells. Primary parietal cells and sporogenous cells. Primary parietal cells going to formation of the parietal cells, and here sporogenous cells, they forms the MMC, megaspore mother cells. Megaspore mother cells undergo the meiotic division. After meiotic division, they form the linear megaspore tetrad. Linear megaspore tetrad. Clear? They form the linear megaspore tetrad. Out of 4, I told such type of structure form, in which 3 become degenerate and 1 become functional. Okay, 1 become functional, and this is called the, this structure is known as the functional megaspore. This is known as the functional megaspore. Okay, which is known as the functional megaspore. Now, so this structure representing, suppose that this is the structure, it is the functional megaspore. Now megaspore enters into the mitotic division. How many times? 3 times. So 1 times they will form the 2, then second time they will form the 3, and third time they will form the 8. So megaspore, this megaspore, suppose that this is the megaspore single, and this megaspore undergo the mitotic first. Mitotic first. Okay, wait 1 second. Just wait. Uh, I didn't use the charger, so we need the battery charger. Okay, wait. Just I think we have only 1 minute. Okay, no issues. because we have very less time also, only 2 minutes. Within 2 minutes your class will be finished, so no issue. Whether it will be finished or not, that not matter, because we have only 1 minute more. Okay, so in this way, if it is going to switch off, no problem, because we have only 1 minute, because after 1 minute your class will be ultimately finished. So we are not going to involve again in charging. Okay, listen. This is the mega functional megaspore. It is the functional megaspore. It will undergo the mitosis first. They form the two, here and here, and they forms two cells and move opposite pole, move towards the opposite pole. Then again mitotic second, then form the four, and they also moves towards the opposite pole. Then again mitotic third, they form the total eight cell. Okay, total eight nucleus, not cell, total eight nucleus, total eight nucleus. And out of eight nucleus, just look it, one will come into center, and from means one from the micropylar end, another from the chalazal end. Both will start to move and come into center. Now you can see one came here, one came from here, and one came from here. Clear? One came from here and one came from here. If one came from here, the remaining three, they will form the antipodal cell. And here one came from here, remaining three, in which one form the egg cell, and the two remaining two, they will form the synergids. Remaining two, they will form the synergids. Ultimately, they form the chalazal end. So here this is chalazal end, chalazal ends, and this structure is known as the antipodal cell. And here this is the central cell or polar nuclei. They have the two nucleus, central cell and two polar nuclei. And here this is the egg cell, and this is the two synergid cell. This is the synergid cell, and one more egg cell. Okay, so this is the overall